It is my honor now to present our keynote speaker, Dr. Slava Epelman from the University of Toronto. Slava is a clinician scientist at the Department of Medicine Division of Cardiology and is affiliated with the Peter Maunck Cardiac Center. He holds key roles as a senior scientist at the Toronto General Hospital, as well as being the Loretta Rogers Chair in Immuno-Bioengineering for the Ted Rogers Center for Heart Research. His research primarily focuses on the role of macrophages in cardiac tissue injury, inflammation, and regeneration. Slava is one of the most world-renowned figures in the field. He has published seminal papers describing the cardiac macrophages, both resident as well as bone marrow-derived macrophages, and the role in cardiac injury as well as cardiac regeneration. His work helps us better understand how immune response can either promote cardiac healing or exacerbate damage after injury. He's deeply involved in exploring how tissue-resident macrophages and monocyte-derived macrophages play distinct roles and how these two major populations of macrophages interact in the heart to promote repair and disease progression. So please join me in welcoming Dr. Epelman to the podium.
Okay, well, thank you for the invitation, Ahmed. It's really great to be here and to be a part of this seminar. I didn't get to know Dan, but sounds like it's just really an amazing group that he brought together. So again, thank you for the invitation. So today I'll talk to you about macrophages and what we've learned from mice, ultimately to help this problem here in the bottom, which is scarred tissue. So I'll share with you three stories. The first are our background, describing our progress in understanding macrophages in a steady state, and then what they do in the setting of ischemic injury and hypertensive injury. And the second part will be a little bit about, we learned about macrophages in the heart. We wondered if the macrophages in other tissues were organized in a similar fashion as they are in the heart, as a starting point to understand macrophage biology. And with that, I'll get into the newest data, which is about our venture into stem cells and trying to engineer these macrophages and combine them with other cell types to make mini hearts, and how this can be a potential future platform to model multicellular interactions, but also a regenerative medicine approach. So I often start talks with this slide. What's similar between a neonatal mouse, a salamander, and a newborn human? So if you amputate the limb of a salamander, that regrows, so that's regeneration. So Nadia Rosenthal's group showed that if you deplete macrophages at that time, you don't get regeneration, but instead you get scar formation. However, if you amputate underneath that scar and you allow macrophages to be present, that regeneration comes back. So macrophages are sort of needed for this remarkable regenerative capacity. And in collaboration with Corey Line and other groups, they've shown that macrophages in the neonatal mouse heart, so in that first day after birth where it can regenerate, macrophages that are there at the time are essential for this process. And if you don't have that, if you deplete those macrophages, you instead form a scar. In Josef Penninger's group, as well as you can study in a newborn baby that had a large embolic myocardial infarction. So despite significant injury there seemed to be a full recovery. So we think all three are capable of tissue regeneration. Mammals lose this ability with age and instead form a scar, and we think macrophages are important in this process. They're not solely going to drive regeneration, but they're key to sort of allowing regeneration to occur in tissues where that is possible. So the conventional view for many decades is that the bone marrow makes all your immune cells. So with respect to macrophages in tissue, the precursor is found in the blood. The monocyte, it comes in and replaces the tissue macrophage. But what we've come to understand is that there's complexity here. Not all of your tissue macrophages are replaced by circulating monocytes. So how do we do these studies? So in the mice, we perfuse the tissue, perform flow cytometry. So here's a standard flow cytometry plot of cardiac macrophages. So we use two markers here, CD11B and CD64, that gets you macrophages. So we used to describe these as M1 macrophages and M2 macrophages, more like inflammatory and reparative, but we and many other groups think this is really an oversimplification of what happens in vivo. Here's what we think about cardiac macrophage origins. I'll just like walk you through this quickly, and you can see up at the top, some of these origins are from embryonic development. And so I'm talking about here an adult, but I'll walk you through time. So in the beginning, the yolk sac, through primitive hematopoiesis, makes macrophages directly without monocytes. These tend to express markers like TIMD4 live 1 folate receptor 2, both in the mouse and in the human. They don't express high levels of MHC2 on their cell surface. So the first macrophage that comes into the murine heart, and we think in the human heart as well, is joined later, a little bit later, in embryonic development from fetal monocytes made in the liver, which we actually also think goes through the primitive hematopoietic pathway originally from the yolk sac. After birth, some of these yolk sac-derived macrophages will upregulate MHC2 on the cell surface. Now, in parallel to this, and even before birth, circulating monocytes from definitive hematopoietic stem cells will enter into tissue. These monocytes express CCR2, chemokine receptor on their cell surface. They'll downregulate CCR2, and they'll become very similar, if indistinguishable, from these other subsets. Lastly, monocytes will enter the tissue. They'll retain CCR2 expression within tissue. And this makes the three, generally speaking, three macrophage subpopulations that we think exist in the murine steady state, and similar populations in the human. The CCR2 macrophages correlate with some inflammatory pathways and also adverse cardiac outcomes in patients where they've been sampled. The CCR2 negative subsets have other functions. They tend to have these sort of phagocytic, endocytic functions. They can promote cardiomyocyte growth, trigger angiogenesis, and are the key cell types involved in the regeneration of the neonate. So we began some of these studies a few years ago, trying to understand the heterogeneity of macrophages in the heart. So Sarah Dick, who has her own lab, and Sarah Najad, a computational scientist, did some single cell work. So we sorted macrophages, which would be found here, and we also have an interest in dendritic cells, which I won't talk to you about today, but we found classic DCs, so these are sort of your DC1s and DC2s, if you know that literature. And we performed our first single cell experiment circa 2018. So here I'll just I'll tell you about the macrophages that we found. So if you look at the macrophages, this is what we observed. So we saw 5 clusters. One is a monocyte, which is just a contaminant, and these other 4 macrophages. So the one at the bottom expresses TIMD4, and you can see here in a pseudotime plot, so this is just change in gene expression as we go across cells. So this is TIMD4 macrophages. TIMD4 macrophages tend to have less antigen-presenting machinery, and this gene H2DMA is one such. What's an interesting relationship, and I'll just again focus on these TIMD4 macrophages. TIMD4 macrophages have high levels of IGF-1, a growth factor. And the other macrophages have high levels of I-1 beta, and you see this sort of reciprocal expression where when you become a TIMD4 macrophage, you kind of gain this regenerative or reparative ability and lose this potential inflammatory component. In the green, we have CCR2 macrophages. We have this macrophage in the middle. This is the MHC2 macrophage. This last one I will not talk to you about, but happy to talk later. It's enriched in interferon-stimulated genes. We and many other groups see these macrophages and other cells, including fibroblasts and endothelial cells, have this signature. We're not 100% sure what it means yet, but groups are working on it. So we can find these macrophages by flow cytometry. So here is just a t-SNE plot of using these characteristics to form this plot. And here are the proteins. So TIMD4, live 1 folate receptor 2. You can use any one of these to find the macrophage. This particular subset, we use TIMD4 just because of its fluorescence profile. The other 2 populations both express high levels of MHC2, so we don't think that's enough to distinguish the 3 populations. You really need CCR2 in there. And typically we use TIMD4, CCR2, and the subset that doesn't express either. Other groups have called these macrophages, at least these TIMD4 macrophages, by other names. So some people call them live 1 macrophages, like I said, folate receptor. So one of the papers, we just called them TLF macrophages for short, like they express either or all of these 3 proteins. So I said to you that monocytes don't give rise to some of the macrophage populations we see in tissue. So how do we know this? So one of the surefire ways to know this is through an experiment where we surgically join 2 mice, a parabiotic experiment where we have chimerism of the blood monocyte. In this case, we link the mice for 6 months, and we often use one animal that's a monocyte deficient animal, CCR2 knockout, and one wild-type animal, and we analyzed the knockout. The reason why is we achieve very high levels of chimerism of the blood monocytes in this situation. So if we look in that red mouse, the CCR2 knockout, all the monocytes in that mouse are from the adjoining mouse. The CCR2 macrophages are replaced. They're mainly entirely replaced. And this occurs quickly. You don't have to wait 6 months. The point here is if you look at the TLF macrophages, these TIMD4 ones, none of them are really replaced. Just a few percent, even after a long period of time. The MHC2 high macrophages, I would say to you, behave a lot like these TLFs or TIMD4s, and not so much like the CCR2 positive macrophages. So there's some replacement, but not nearly what you get in this monocyte-derived subset. In that study, we were asked, well, it's 6 months. How do you know if you just don't keep on waiting and you go into aged animals? That doesn't change. So we did it a bit longer, not truly aging studies where they're 24 months of age, but we compared parabiosis at 6 and 10 months. And this is total chimerism of cardiac macrophages. We don't really see any progressive replacement similar to brain microglia or alveolar macrophages. If you go by subset, again, the TLFs have virtually no contribution of monocytes to them. So we think these three populations have different life cycles in relationship to blood monocytes. We also see some conservation to human. So the first is visually. So if you look in the human, and at this time we had a few populations, if you look at the CCR2 negative population, the human is TLF macrophage here. It's got these large vesicles, and it looks a lot like this mouse TLF positive population. Similar in transcriptome in terms of endocytic machinery. The CCR2 macrophages in both are enriched in some, but not all, inflammatory pathways. One pathway that stood out to us was the IL-1 beta pathway and the NLRP3 inflammasome. The CCR2 negative populations tend to have growth factors, and here's an angiogenesis assay tube formations. If you take the supernatant of these two predominantly embryonically derived subsets, the TLFs or the MHC2 high macrophages, you put them with endothelial cells, they make tubes. The CCR2 macrophages, they don't make tubes. And this is dependent on some growth factors like IGF-1. In our lab, we use a variety of genetic systems to track macrophages, a lot of Cres, some constitutive, some inducible. And the point of these studies is to try to separate out the cells that live within the heart before you injure it versus the cells that come to the heart after you injure it. So I'll just tell you, summarize the data with these two forms of injury, ischemic and hypertensive stress. And this is the workhorse in our lab. We use this animal, it's a CX3CR1 Cre-ER, so it's tamoxifen inducible, and the ischemokine receptor is expressed on macrophages and monocytes broadly. And when we give tamoxifen during the pulse, the macrophages and monocytes all become red. In this case, it's a ROSA TD reporter and also a diphtheria toxin receptor, so we get both of them. So the diphtheria toxin receptor is expressed on the cell surface. We remove tamoxifen, and this tamoxifen is given usually at the time of weaning, so around 3 weeks of age. So we give them a week of tamoxifen in a chow, we take it away, we come back to these animals at 8 to 12 weeks of age. So at that time, really, it's only just the resident macrophages that were labeled at the time. Monocytes are no longer labeled. So if you give diphtheria toxin, you're just killing the macrophages that were there or tracking them. So this is like I said, the disease. And if you take the macrophages from this infarct area, so this is a complete infarct in a mouse, and this is the labeled resident macrophages. Within this ischemic peri-infarct zone, there's a total reduction of macrophages. They do proliferate at the border regions and increase their numbers just at that one spot, but overall there's a reduction, and you can see numerically how many monocytes that would become macrophages come in. So here you're 160 cells per milligram, and here you're about 5,000 cells per milligram. So you can see the sort of magnitude of the cells and their numbers. In this system, we specifically depleted the resident macrophage. And what it did not change was it did not change the infarct size. It did not change the ejection fraction or scar size at 7 days post-infarct. But what it changed was cardiac function from day 7 to day 28. So a little bit of infarct expansion into that peri-infarct zone and remodeling changes there and some LV dysfunction. So we think, despite them being depleted already with the injury, the remaining cells do have a functional benefit. We'll change gears here and tell you about a different study. So hypertension, you know, we've heard about HFpEF, but certainly hypertension is one of the main risk factors for heart failure and cardiovascular disease. It affects 1/3 of the world's population. And we were interested in the very earliest parts of this, so an adaptive phase. So when the heart first faces hypertensive stress, there, we think, is an adaptive change in the size structure of the heart, ultimately becomes maladaptive. And this growth, we were wondering, does this sort of replicate what happens in development in the regenerative window where macrophages have been studied? So we modeled this. We didn't do it with TAC, we did it with angiotensin II infusion. So if you infuse angiotensin II alone with pumps, the blood pressure goes up and you get hypertrophy and some fibrosis. And we wondered whether the resident macrophages were involved in this process. So first, I'll just summarize the data, but what we found in our genetic model is when we depleted the resident macrophage, this protein IGF-1 was reduced in the steady state. So about 25% of it was made by resident macrophages. In the setting of hypertension, the IGF-1 protein goes up, but if you deplete resident macrophages, it no longer increases. But there's still IGF-1 protein present. So we think resident macrophages make some IGF protein, IGF-1, and that increases with hypertension. So student Raisa, who's now a postdoc in New York, she deleted IGF-1 in that system just within the resident macrophages. And what we found was that this sort of heart weight to body weight, this sort of increase in mass of the heart that you see with angiotensin II, this was blunted if you knocked out IGF-1 in the resident macrophage. And if you actually look at the cardiomyocyte area, it's more striking. So in sham here, going with angiotensin II, you see the increase in the cardiomyocyte cross-sectional area. So they're getting bigger. And they do not hypertrophy when IGF-1 is removed. So it's a bit different sometimes in the studies that we've heard about today, where you're exposed to hypertension and the LV function is preserved when you do something, and the cardiomyocytes don't get big. And so all forms of hypertrophy are not the same. We think there's aspects that are more adaptive, and we think aspects are maladaptive, but yet the cardiomyocyte is bigger. So in this instance, we think that this growth is an adaptive growth. We don't really see much of a change in LV function. We just see that growth, and this is what I'll show you here. So if we look at LV ejection fraction, there's this very modest decline, or sometimes we don't see much of a decline in the wild-type animals, but there's this further decline if you knock out IGF-1, and that's sort of correlated with an increase in end-diastolic volume. So in this study, we had deleted IGF-1 from resident macrophages, and this was the phenotype. LV dysfunction, failure of the cardiomyocytes to grow, ventricle dilates. When we kill the macrophage, instead of just deleting IGF-1, when we kill the macrophage itself, the only difference is we also see fibrosis. But we don't see any excessive fibrosis here with just the loss of the growth factor. Okay. So we saw these macrophages, and then we thought, well, this is the way they're organized in the heart, and they sort of behave in certain ways. And we wondered, does this extend to any other tissues in the mouse? So we sort of embarked on harvesting every organ from the mouse. We profiled 5 organs more deeply and superficially, essentially all of them, to see if this structure of macrophages was conserved in some way. So Anthony and Sarah Dick were involved in this study. So this is just flow cytometry of some typical organs you might see. So liver, heart, lung, kidney, and brain. I'll just caveat this by saying in the lung, we did not, these are just the interstitial macrophages and not the alveolar macrophages. In the brain, we're not talking about microglia. We're talking about these sort of border-associated or perivascular macrophages in the brain. So there's this sort of similar flow cytometric pattern, we thought, in these organs. So we thought perhaps these markers could stratify different populations. And then so we performed single-cell sequencing on those 5 organs. And so here at the bottom is just a representative example of TIMD4 macrophages at the bottom and CCR2 macrophages at the top and MHC2 in the middle. And so we wanted to know whether the same genes are present in the same macrophages across organs. And so we looked at their conservation. So what I'll tell you is if you look at TLF macrophages, 75% of the genes across these 5 organs are organ specific, so that the organ instructs that macrophage to have certain functions. And most of what makes a TLF macrophage in one tissue is not the same thing as it is in another tissue. About 11% of the transcripts were conserved in 3 or more tissues. And it's sort of declining in order after that. So CCR2 macrophages are next, being the most conserved at 9%, and the MHC2 high is really the most organ specific with very little overlap. But if you look carefully, across these organs. You can find some signatures that are similar for organs, the TLFs being the most conserved, and the others having a more of a loose conservation in terms of transcripts. So Anthony and Sarah went on and they said, well, we see this in these five organs. What about all the other tissues? So they studied fat, brown adipose tissue, white adipose tissue, various skeletal muscles, the tongue, digestive organs, skin, uterus, testes, etc. So by flow cytometry, you can kind of see similar populations. And then they wondered, well, is the lifestyle or the life cycle or relationship to monocytes also similar? Are some monocyte dependent and some not in these different organs? So we did a couple of different studies. One was parabiotic and the other was fate mapping. And I'll just summarize here the parabiotic studies. So this is just a heat map looking at all the different organs and the subpopulations. And red means it is that subset of macrophages was not at all replaced by monocytes. And blue means, like really blue means it was 100% replaced, just in this very short 5-week experiment. So you can see that in all these organs, the TLF macrophages really were not that well replaced by monocytes, and the CCR2 macrophages were. And the MHC2 high were kind of in the middle. And our genetic fate mapping data sort of supported these observations. So what I've shown you is we thought that there was this loose conservation, that we thought this was a good place to start if you're studying macrophage biology. You know, like what markers should you use? Like why pick these over those? So we thought that this framework at least kind of tells you the life cycle of the macrophages that live there in the steady state. And in inflammation, you know, things can get a little bit hairy. So, but in steady state, we think this is a good place to start. The TLF, like what I didn't get into here is that the TLF were by far the most conserved. So they're the most conserved across organ. They're the most conserved when we compare it to human. And we wondered, well, you know, if they're so conserved, maybe we should make them. Maybe we could exploit them as a potential therapeutic strategy. And so here I'll change gears and tell you about a different story. So this is the problem, and one of the ways that folks have addressed this problem is making a lot of IPS or ES-derived cardiomyocytes, differentiating them in vitro, and then injecting them into the scar tissue. We have several investigators at my institution that do this, Mike Laflamme. So billions of cardiomyocytes go in, and they've achieved some interesting success with partial remuscularization of that scar tissue. One of the problems, though, is electrical integration. Some of these animals suffer from dangerous ventricular arrhythmias. And so we wondered, you know, given these issues, perhaps, you know, additional cell types that are important could help, and in this setting, could TLF macrophages help? So one of the students in the lab, Homaira, undertook this project. And here's just some single-cell data of the human heart. We get some pediatric samples from our Sick Kids Hospital. And so one of the populations of macrophages from these two patients were these sort of live one folate receptor 2 macrophages. What I'll tell you interestingly is even though we use TIMD4 in the mouse exclusively almost, TIMD4 is not really expressed in most human macrophages. It's on in development in every macrophage, and it sharply is downregulated in most macrophages. So if you're going to study human, live one is a better marker. She actually found another data set that had a variety of animals. This was, I think, for COVID. So this is cardiac macrophages from these animals, and she looked at this TLF gene signature, and then across these animals, they all had this TLF signature, suggesting, you know, beyond mouse to human, many organisms have this. And so we do rely a lot on publicly available data. So this is from the human fetal cell atlas. So this is 4 weeks post-conception of the yolk sac, sort of the contents and the membrane. And if you look, and these are all myeloid cells essentially. And basically all these macrophages are the live one TIMD4 folate receptor 2 macrophages. The yolk sac macrophages in the human all express these markers. And so we thought, okay, let's go and try to make this. So we started this collaboration, and it was great until COVID hit, because it's like a 4-way collaboration. So so Mike Laflamme is a great stem cell biologist, and he's the one who implants all those cardiomyocytes into the scar tissue. And Gordon Keller is a stem cell biologist that can make any type of stem cell. And he actually had a system where he could specify a primitive hematopoietic macrophage. And Milica Radicic is a talented bioengineer that makes these little mini hearts. And let's see if this will play. Don't quite have my pointer. Let me see. I might not get it to play. There, beats. So basically, these sort of tissues are cardiomyocytes. So you can purchase, make your own cardiomyocytes, add fibroblasts, and they condense into this muscle fiber, and it pulls along the wire. And you take movies with a camera, and you can do a lot of measurements on the systolic diastolic function. So we worked with these groups together to try and understand if macrophages could be helpful. So Gordon's protocol for making macrophages is published, and we helped him characterize this. So what happens at the end of about 2 weeks, not that long, is you have CD45 positive here in pink, CD14 positive, CD64 positive macrophages, so they're typical macrophage markers, and then they express LIVE1 and they don't express CCR2. So these are the macrophages that we were using, human ES-derived macrophages. And this is the way that it looks. So this is the microtissue, these little mini hearts. So the standard was the cardiomyocytes, so ES-derived cardiomyocytes, or IPS, fibroblasts, which are actually primary human cardiac fibroblasts, and then we compared addition to a macrophage to not. And macrophages in this case express RFP. So this is what it looks like when you first seed it in a hydrogel matrix, and then very quickly it condenses down into this fiber. And what we've actually observed within the first day, this is day 7, is that the tissue is thinner. So it's sort of the macrophages, even though there's actually more cells in the macrophage group, we kept all the other cell type numbers constant, just added more macrophages, the tissue is thinner. And it's an observation that a couple of groups have seen. So Florent Genoux did a similar thing where he had IPS macrophages and specified them into microglia. And in the brain organoid model, it also sort of reduced the size of that organoid. So it seems that the macrophages control the size of the tissue. And we're immunologists, so the first question we actually asked is an immunologic question. We wondered, this is a macrophage we made in a dish. How similar or different is it to a human macrophage? And are these macrophages, taught anything when they're in the tissue? So we actually do think they go to school. There's just a few genes, core cardiac macrophage genes of the macrophages just cultured in the hydrogel matrix alone, cultured with fibroblasts, and with the fibroblasts lay down a lot of collagen, and it looks the same as what you just saw. It just doesn't beat. And then the 3-cell combo. So in the 3-cell combo, we really saw a strong upregulation of a core macrophage gene signature. In particular, these genes, LIVE1, folate receptor, MRC1. What really stood out, though, in terms of the pathways, the main pathway that was induced in the macrophages with this pathway related to efferocytosis. So fibroblasts induced it, but it was further induced with the cardiomyocytes. So efferocytosis is a process where a macrophage eats stressed or dying cells or material. These macrophages, when they were educating these tissues, looked a lot more like human macrophages based on their gene score, and this was the main program that was upregulated in the macrophages. So functionally, adding the macrophages is indeed important. So the active force of contraction increases when you add macrophages. The rate of relaxation increases. So you have this sort of improved systolic diastolic function. And you have increased conduction velocity and increased calcium amplitude. Another interesting observation I put out sometimes if I'm talking to a cardiovascular group is that we did a lot of experiments with different preps. Some of them were commercial, some of them we made ourselves. Like I said, ES or IPS, and this occurred over the years. And basically, sometimes we would get a prep of cardiomyocytes, and their intrinsic depolarization rate was like 30 beats per minute. And at other times, we would get it and it'd be 130 beats per minute. So they're very different from each other. And so this is just a curve showing the relationship between the starting heart rate without macrophages to what happens when you add a macrophage. So on the x-axis, you have that starting heart rate. So this particular prep on the bottom, we got it was 130 beats per minute. We added macrophages, it decreased the heart rate by about 50 beats per minute. Whereas those at the top, it increased the heart rate. So there was a sort of reciprocal effect depending on what the starting heart rate was, kind of sort of bringing that heart rate to sort of more towards the middle. At least that's what we thought. So it very much changed that electromechanical coupling of these cells. We did a lot of experiments to try to understand what was happening. We did a lot of transcriptomic work. We didn't really get too far with bulk RNA or single nuclear RNA-seq. And so we actually moved to proteomics. We did proteomics of the individual biowires, or microtissues. And the main pathway that came out with respect to the cardiomyocytes is the upregulation of sarcomeric proteins when we add the macrophages. So very quickly at 3 or 14 days. And we even saw isotype switching from the fetal to adult isoforms, indicating that more of this contractile machinery was being made and it was seeming to become more mature. We also observed that very rapidly the macrophages changed the microenvironment. So even in the first day, we saw this blunted release of mitochondrial DNA. We saw decreased tissue ROS, decreased release of CK-MB, and LDH as your standard marker of cytotoxicity is decreased with the addition of macrophages. So macrophages are supporting this sort of healthier environment right from the beginning of culture. They reduce the rate of apoptosis of the cardiomyocytes. And the part that we are interested in is that what I mentioned earlier, efferocytosis, so the eating of stressed or damaged cells. So here, Homaira labeled cardiomyocytes one color, and then the green in this case was CFSC dye, and all that green dye, a lot of it ended up in macrophages. So the macrophages became very, very green, and we could interrupt this process by adding simply just by adding excess annexin. So annexin binds phosphatidylserine that's translocated to the cell surface, not just in dying cells, but stressed cells. And by interrupting that, we actually prevented the macrophages from sampling that material. And when we prevented that sampling, we prevented the benefit. So this is just the LDH release. So in gray is where it starts, and then in red is where macrophages improve it to, and that's blocked in the hatched, indicating that if macrophages can't sample the material, they can't provide that benefit. And that translates into blocking that active force that's induced by macrophages, as well as the contraction slope, so the rate of contraction and the rate of relaxation. Okay, so the macrophages are sampling, they're eating, but like what does that actually do? So some of the results were a little confusing to us. So I'll actually start with this, which is the last experiment that we did. When we looked at the total amount of ATP in the microtissues, there was more when we added macrophages. So this is just ATP stores. It's not seahorse or flux. Okay, so the main pathway that was down when we added the macrophages in the microtissues were mitochondrial pathways, which is a little bit opposite to what you would think with cardiomyocytes. We're saying we're differentiating, they're becoming more mature, so probably they have more mitochondria, but it's actually not the case. So if you look at ATP synthase, mitochondrially encoded, ribosomal, or structural proteins, these aren't genes, these are proteins by mass spec. They are all markedly reduced by the presence of macrophages. So we thought, oh, that's confusing. And if we look carefully, though, if you look at the TCA cycle, I didn't realize this because I forgot this biochemistry. So there are some isoforms that are in the cytosol and some isoforms that are in the mitochondria. So all of the mitochondria localized proteins that are part of this pathway are reduced, but the cytosolic ones are increased. So we started thinking, well, maybe just the mitochondria are gone. It's not so much that the pathway is different, that the physical mitochondria and all the proteins that those mitochondria contain are being eliminated. So that was one thought we had in our head. So we thought, well, what happens if we interrupt this eating? What happens? So this is mass spec of some of these mitochondrial proteins. And we have cardiomyocytes and fibroblasts on the left and with macrophages on the right. And this is just adding the annexin. So blue is the down regulation you see with the addition of macrophages. And if we stop the macrophage eating, we no longer see that reduction of mitochondrial protein content. So that eating, the macrophage sampling of the material is required for this. And when we block that eating, when you look at the TCA cycle, these proteins that are mitochondria localized are no longer reduced. Interestingly, though, those that are induced in the cytosol, the eating has no effect on those. So we think that there's a sort of a compartment-specific effect, and we actually just track the mitochondria themselves with MitoTracker, and indeed the mitochondrial abundance within the cardiomyocytes. We have all the same kind of parallel data in the fibroblasts, so I won't get into it today, but the same thing happens with the fibroblasts that are in culture. The macrophages are also reducing the mitochondrial content of the fibroblasts. So we got asked a lot of metabolism questions on that paper, and so we performed CyTOF. We have a 36-marker CyTOF panel that includes metabolic proteins. And so this is a CyTOF UMAP. So on the left is just your cardiomyocytes and fibroblasts, and on the right is addition of the macrophages. And the macrophages are these ones here in the blue. And this is like the QC step. So the very first thing that we noticed is these dead cells, what light up with the dead dyes, are markedly reduced with the presence of the macrophages. And these dead cells here also contain ATP5A, which is a mitochondrial protein. So this is just the reduced, excuse me, the number of events, the reduction in that number of events by the presence of the macrophages. So that's sort of the first step. So we're seeing that these sort of debris and or dead cells that contain still contain a detectable mitochondrial protein are really reduced by the macrophages. And if you stain for it, you can see it here. So the troponin here is in purple, and ATP5A is in green, and this green is sort of within and without, like outside of the cells, and all of that content is really reduced by macrophages. If you look actually within the cardiomyocytes themselves, we saw some interesting things. So with these 36 markers, you could say that the cardiomyocytes clustered made 4 clusters. Cluster 3 and 4 were similar between the two groups. The greatest difference we saw was sort of cluster 1 and 2. So cluster 1 seemed to be present just in the cardiomyocyte fibroblast group. When we added macrophages, cluster 1 sort of ceased to exist, and you saw cluster 2 form. So what's the difference between the two? It's not exactly clear, but it seemed like there was downregulation of mitochondrial pathways, so actual proteins related to mitochondria within cardiomyocytes, so ATP5A, and other citrate synthase, and you saw potential upregulation of glycolytic pathways like GLUT1 and MCT1. I won't show you this here, but we actually looked at single-cell data from humans across different times post-conception, so 5 weeks to 25 weeks. It was a previously published study. So like the authors found, we also found interestingly, a downregulation of oxidative pathways in early human ventricular cardiomyocytes. This is pre-oxygenation stage. This is just in development. So we think the macrophages sort of kind of help guide the cardiomyocytes to a less dangerous, oxygen-dependent pathway where there could be more ROS under these stressful circumstances to more glycolytic. So to summarize, this last part, we developed these, I guess we call them immuno-engineered tissues, where macrophages that are essentially tissue culture macrophages really look a lot more like fetal macrophages once they're in the tissue for a period of time. This is very much an interest of ours to understand how these macrophages are programmed to become tissue macrophages. What are they eating? Is it different types of cargo? There's cardiomyocyte, there's fibroblast. There's also the different organelles. So Andreas Hidalgo and his group published a nice paper where cardiomyocytes in the mice expel damaged mitochondria and macrophage in exopheres, these membranes, and ingest it. So we think something similar is happening here with macrophages sampling or eating these sort of mitochondrial components and mitochondrial debris. And this is part of the education. So macrophages are also functionally important. They improve contractile and relaxation properties. And they also reprogram, like I was saying, the ventricular cardiomyocytes that are ES-derived to be more in vivo-like, at least on their transcriptome. Sorry. And ultimately, this process is dependent on eating. So with animals, our next step is to inject these into animals. So lots of cardiomyocytes, lots of macrophages, lots of engineering work to go, though, still. I'll just end by saying that all of our data is available on our website. So if, for example, you wanted to look at the proteomic data, so this is mass spec, day 3 with and without macrophages, day 14 with and without macrophages, you can just type in your favorite gene of interest, and you can look. And so it's all up there. You don't have to download it and have your bioinformaticist do it for you. You could just do I do this actually when I'm listening to the talks today. I kept on searching the different proteins being mentioned to see if they were differentially regulated in our tissues. And with that, I'll end by saying thank you to the people that did the work. So Homer is a talented graduate student who did the last part of that project. Anthony really contributed to the pan-macrophage work. Sarah Dick is a former lab member who has her own lab. And I'm happy to take any questions. Thank you.
I can start. I have a couple of questions. I'm very interested in the MHC2 positive macrophages, and they are living there between the TLF and the CCR2 positive cells, do you think they are originating to some extent from the TLF macrophages, or are they related functionally to the TLF macrophages?
It's a good question. So, I mean, we actually didn't want to call them MHC2 macrophages because they're not. It's not really a very specific name because the other cells also express MHC2. We actually wanted to call them double negative macrophages, but we weren't allowed. We had to change the name. So, if you look before birth, they're there. So, before birth, you have CCR2 macrophages that were just monocytes, probably like 6 hours before that. And then you have the TLF macrophages, and you have that subset of macrophages that's in the middle. From our perspective, they seem to be present almost at the same time in the neonatal heart, in the murine neonatal heart. So it's hard for us to know if one comes from the other. But yolk sac macrophages express those transcripts and those proteins. So TIMD4 live 1 folate receptor 2 in the mouse. And then yolk sac macrophages, as macrophages, migrate into tissues that are available even before the vascular system is formed, including the heart. So there's sort of two options there. The TLF macrophages that came from that yolk sac at that time downregulate that gene program and become this other cell. When we fate mapped E7.5 to E8.5 macrophages, like labeled what was around there with what we can do, and we looked in the adult, we labeled both the TLFs and these MHC2s. They both come from the yolk sac. So that interrelationship just needs another tool. Like we need another tool to know if one truly originates from the other or they're originating simultaneously. So it's hard to answer that one.
The other question is related to, how macrophages control the metabolism of cardiomyocytes. That's quite interesting, especially during the developmental stage in the first week after birth, and how cardiomyocytes shift their metabolism. How much of this could be related to the macrophage infiltration into the tissue from bone marrow and their function?
Well, in vivo, hard to know. I mean, we have mice models that lack macrophages during development, so we could test that. So my lab mate, Corey Levine, published a paper where some macrophage deficiency in neonatal development leads to altered vascular patterning. I mean, those mice have a lot of problems. These are like OP mice and sort of like a number of problems. But immediately after birth, it's a vascular patterning issue that was the most obvious, but metabolism never studied. I mean, they have hearts. Their hearts form. In our system, we think the effect is both a direct cell-to-cell effect, so the eating, obviously taking away all that mitochondrial content, and that changes the environment. Now you have a whole new environment, and then I think the cardiomyocytes are behaving differently. I don't know how much the macrophage is shaping the subsequent environment itself. There's lots of cytokines that the macrophages here are making that are specific. Some cytokines that were high before the macrophages were added go down. Others that weren't present, like IL-10, are now up. So there's lots of potential paracrine mediators that could change the metabolism of the cardiomyocytes that may be directly or indirectly from the eating. But there's probably, like I showed you there with some of the mass spec, when we interrupt eating, some of those metabolic patterns are not changed. So some other effect macrophages are having. In vivo, a little trickier to sort out as well.
Terrific, really terrific talk. So do T cells participate in the education of macrophages? You said it was tissue specific, and then I have a follow-up question if they do.
Do T cells participate in the so we never tested whether a T cell in the steady state participates in that education program. We think that the education program is dictated by the length of residency. So the longer you're there, the more time you have to adopt that tissue-specific signature. So a short time, you'll partially adopt it. A long time, you'll more fully adopt it. And I think that's governed probably by more so by the organ itself rather than another immune subset like a tissue-resident T cell that would live there.
Okay, thank you. One of the reasons I'm asking is when a pediatric heart is transplanted, often there's a thymectomy done at the same time. But those hearts seem to know how to grow to the adult size. And so I'm just wondering if macrophages are controlling the size development of an organ, how does a pediatric transplanted heart know to grow how big it should?
That's a whole different, we've talked about that before, from a macrophage perspective, a solely, not organ, but solely macrophage perspective of quorum sensing. How do you know how much you have of a certain cell type? Like, what is the inhibitory boundary of growth? In the liver, I know when you resect the liver, there's that regenerative capacity, and it builds little units simultaneously. Like you have the sinusoid, you have an actual Kupffer cell and other cells, and it just builds the units one at a time until it somehow knows to end that process. Like, who knows why it ends? We don't know what that boundary, how that boundary is sensed. So I don't know within the heart why that is. I don't think that it's solely macrophage. Like I said, in the OP mice, which lack macrophages, the hearts are, roughly speaking, the same size. They're not really grossly large or grossly small. So that organ probably is just evolutionarily understands how big it should be at that stage. So that was a non-answer, but sorry.
Yeah.
Hi. Hi, thank you very much for your talk. Actually, my question makes more sense if you, the cardiomyocytes that you used derived from the IPS, were they chamber specific or not?
So we've used ES-derived and IPS-derived separately, and they're both ventricular cardiomyocytes, fairly pure.
It was a ventricular specific
Yes.
protocol.
Yeah.
So I have a very naive question about two different points that you made in the first half of your talk. One was regarding TLFs being present and improving tolerance or repair to cardiac injuries. The other one, the obvious one, that regeneration goes down with time. So when do TLFs get educated? What are the processes, and is there enough known about the education to potentially provide therapeutic approaches to improve TLF formation?
So we don't know if the TLFs need. So one of the aims is to determine whether a pre-education of the macrophage is needed before being transplanted with other cells. Like, does it actually need? It is educated within a tissue. It is quite different by hundreds of transcripts being in that tissue just for a couple weeks. But whether that actually aids in engraftment of cardiomyocytes in an animal, so the models like immunosuppressed pig or a nude rat. Right, so does that education program ahead of time aid in the engraftment? I don't know, that's one of our questions. I think the education is just by, eating cargo is a huge metabolic load for a macrophage. It doesn't just take it up, it actually has to digest it and then deal with it. And that very much reprograms the macrophage, apart from anything else, just eating. And the different things that it eats, we think reprograms it in different ways. So we're trying to tease that apart. We'll have to test the experiments. I don't remember the second part of your question. That's good.
Slava, that was terrific. So as I'm sure you know well, years ago, Matthias Nerendorf published this observation that macrophages coupled through connexin 43 with cardiomyocytes and control electrical activity. I'm curious whether you see any evidence of that in your iPS cell systems, whether it's specific for certain subsets, and whether that accounts for some of the electrical interactions that you see.
We think maybe. We have no data for this. We think that maybe we could test this hypothesis in the system at a much later point. This is sort of organization, and we don't think it's acting like a sink, like he had shown, it was on pacing. It was a very subtle phenotype in a very well-formed myocardium at the AV node. But in our model, it's sort of like it's such a difficult environment. Weeks later, these things look great. 6 weeks later, cardiomyocytes are all elongated. And then at that time, what we're trying to build the capacity to do is inducibly delete genes in these models after the tissue has formed. Because you cannot separate out an altered microenvironment in the beginning to all the subsequent downstream effects you may see. So that if the microenvironment's the same and we deleted the gene after everything was well formed, that's where we could probe this connexin 43 question.
I may have a very naive question about the macrophage. So when you stop macrophage from eating, of course we are preventing it from dealing with the huge cargo that it has to process and digest. But then a lot of energy is also coming from there. So what is exactly that macrophage doing at that stage? Do you find that preventing them from eating makes them change in some way? Of course, you haven't seen any metabolic parameters that are being changed, but I was wondering if it changes its intrinsic property like inflammatory cytokines or anything, such kind of things.
So we're studying that right now. In terms of inhibiting eating, so annexin is an interesting thing. You know, we inhibit eating, but we kind of inhibit many types of eating. So our interest is maybe to try, we kind of pause the annexin experiments with the idea of inhibiting or targeting receptors. Like, you know, if it's eating via TREM2 or it's eating via AXL or TK, like, is it eating different things? And then can we reconstruct it that way rather than the whole eating situation? From the mass spec, we have proteins only expressed by macrophages. So we've not done any specific sorting. So we have certain proteins that are exclusively macrophage derived. When we inhibit eating and we look at mass spec, we did the mass spec with and without the inhibition of eating, we see that those proteins, some of which are related to the education or the adopting that tissue resident phenotype, they're blunted. So that I guess what I'm saying in other words is that the macrophage eating is part of the education process, as we can tell indirectly. The other part that I didn't tell you or didn't come out enough is when we block macrophage eating, the changes in the types of collagens that we see laid down is not affected. And when we block macrophage eating, the marked upregulation sarcomeric proteins is only modestly affected. There's a big effect on function. There's a big effect on mitochondrial debris and certain outputs, but there's a whole bunch of outputs that are not that much affected when we inhibit that eating. So there's probably all sorts of other things the macrophages are doing beyond just simply eating the debris. Okay, well, thank you very much.